Today, we are staring down one of the most persistent, really fascinating paradoxes in modern science. We are diving deep into the organism C. elegans.
The nematode worm.
Exactly, the free-living transparent roundworm, and specifically the decade-long international effort known as OpenWorm that has been, well, struggling to simulate it.
And here is the core conflict, the thing that makes this whole story so compelling. C. elegans is famously the simplest animal for which we have the complete anatomical blueprint. Every single neuron is mapped.
Every single one.
Yet this creature, it's smaller than a grain of rice with only 302 neurons, is resisting complete digital emulation. Some of the best computational neuroscientists on the planet are stumped.
Right, and that simplicity is exactly what highlights this massive uncracked problem in how we understand emergence in biological systems. It was chosen for that reason.
It was supposed to be the easy one.
It was supposed to be the solvable one. Back in 1963, Sidney Brenner, the Nobel laureate, he proposed it as the model organism.
Yeah.
He basically bet that its tiny size and fixed cell lineage would let us trace the entire path from genes all the way to behavior.
And for a while, that bet seemed to be paying off, especially with one huge data point.
That's right, the big one. In 1986, John White and his colleagues published the first comprehensive wiring diagram of its entire nervous system.
The Connectome.
The full anatomical connectome of the adult hermaphrodite. So that map, it details all 302 neurons, every chemical synapse, every electrical gap junction between them. We know the entire roadmap.
But we still can't predict the traffic flow.
Exactly. And that's the central enigma OpenWorm was founded to confront back in 2011. If you know the entire wiring diagram, why can't you build a perfect digital copy?
So what was their mission exactly? What was the goal they set out?
It was really explicit. To create the world's first whole organism in a computer through what's called bottom-up systems biology modeling.
Bottom-up meaning build it from the parts we know.
Precisely. And their starting goal was very focused and pragmatic. They wanted to simulate the core locomotor system. So those 302 neurons and the 95 muscle cells that they connect to.
So they weren't trying to simulate everything at once, not egg-laying or finding food.
No, just the most fundamental action.
Yeah.
Crawling.
And this focus is really crucial, isn't it? The challenge wasn't just, counting the parts or mapping the structure.
Yep.
It was about modeling how that complex macroscopic behavior, like the smooth sinusoidal crawl of the worm, how that emerges from all these tiny biophysical processes happening at once.
And in a closed-loop system. That's the key, where the nervous system is constantly getting feedback from the moving body and from the environment it's moving through.
That concept of the closed loop is where it all gets incredibly complex.
Where the complexity explodes, yeah.
Yeah.
The worm is moving against something, usually a fluid-like medium like agar. And that physical interaction, that pushback, it feeds right back into the neural circuits.
So the choice of the worm, C. elegans, was strategic.
Highly strategic, because its known structural base isolates the scientific problem perfectly. If the simulation fails, we know the failure is because of some unknown functional parameter or an error in how we couple the different scales together. It's not because we're missing anatomical connections.
To really get why this is so smart, we should probably compare it to some of the other bigger brain projects out there, like the Blue Brain Project.
Yeah, the Blue Brain Project, or BBP, founded at EPFL in Switzerland. It pursued a totally different kind of complexity. Their goal was monumental.
What were they trying to do?
Detailed reconstruction and simulation of a mammalian functional unit, specifically a rat neocortical column.
Okay, so how many neurons are we talking about there?
About 10,000 neurons in a rat column, which, sounds like a lot, but it's far fewer than the 60,000 in a human column.
But the structure itself is just a whole nother level of complexity, and it's not fully mapped like the worms.
Not even close. So Blue Brain was grappling with both this immense volume of cells and a lot of unknown anatomy. OpenWorm did the opposite.
They went to the other end of the spectrum.
Exactly. By picking the 302 neuron C. elegans, they completely eliminated the problem of network discovery. The map is known. So if they failed to simulate its behavior, the conclusion is, well, it's pretty profound.
That the connectome, the wiring diagram alone, is just not enough to explain life.
It's profoundly insufficient. They isolated the problem of structure versus function in a way no other project really could.
Okay, let's unpack this. We're moving into the blueprint of this digital nematode. If you start a project like this in 2011 with this goal of bottom-up simulation, how do you even begin? How do you manage the data and build the software to integrate all these different scales of biology?
You start with what's called the foundational data stack, and thanks to decades of C. elegans research, it's unparalleled. The full structural map, the connectome, is the centerpiece. That just removes the whole challenge of network discovery right off the bat.
So they had the wiring, but you need more than just wires to make a virtual organism, right? It can't just be a static diagram.
No, absolutely not. It requires integrating several key data components. You need a three-dimensional anatomical map, what they call the virtual worm, which defines the physical shape and location of every single cell.
Okay, so you have the shape and the wires.
Right. Then you need a complete curated cell list, and, critically, you need quantitative physiological data on all the ion channels the worm uses.
That's like the power supply and the control switches for each neuron.
That's a great way to put it. And finally, for validation, to check your work, you need massive databases of real-world worm behaviors, phenotypes, things like the worm behavior database.
To benchmark your simulation against what a real worm actually does.
Exactly. The target behavior repertoire is pretty expansive, finding food, laying eggs. But the first functional goal, the absolute first step, is simulating simple motor response, especially that classic sinusoidal crawling motion inside a virtual environment.
And this brings us to what you said was the core technical hurdle they had to solve, closing the neuromechanical loop.
Yeah, this is the big one. This integration challenge hadn't really been solved by any previous nervous system model when OpenWorm began.
So let's visualize this loop for a second. It's this constant conversation between the brain, the body, and the world.
It is. It starts with an environmental stimulus. Let's say the texture of the agar plate or the smell of food.
That sensory data gets converted into electrical signals. That's sensory transduction.
Mm-hmm. And that signal then runs through the network, triggering interneuron firing, and then eventually motor neuron firing.
Okay, so the brain has sent a signal to the muscles.
Right. And that motor signal translates into physical action, motor output.
Yeah.
This causes the worm's soft, elastic body to deform body deformation, and it pushes against the environment.
And here's the crucial part, the step that makes this different from just an abstract computer model. That physical deformation, that bending and squishing of the body.
Generates proprioceptive feedback.
The body is sensing its own position and movement.
Yes. And that feedback signal loops immediately back into the neural circuits, affecting the very next round of firing. It's a constant, instantaneous cycle.
And that integrative approach is so critical because the worm's movement isn't just. It's not like a pre-programmed robot arm going through the motions.
Not at all. It's highly dependent on the environment and the mechanical squishiness of the body. If your virtual worm is too stiff, or if the fluid dynamics of the virtual agar are wrong, the proprioception will be wrong, and the entire neural circuit instantly breaks.
The moment they realized they had to model this entire closed loop, this multi-scale coupling, they must have known that a traditional single algorithm simulation just wasn't going to cut it.
They knew. You couldn't build a monolith, a single piece of software to do everything. They needed a modular, multi-algorithm architecture.
And that's where their custom-built platform, Geppetto, comes in.
Exactly.
Yeah.
Geppetto is this reusable, open-source, web-based platform that acts as the operating system and the visualization hub for the entire virtual organism. It's the middleware that holds all the separate pieces together.
So for those of us who aren't software engineers, why was abandoning that monolithic approach so important for this kind of science?
Well, if you build a monolith, a single, tightly integrated piece of code, every time a scientist makes a new discovery, say they find a new variant of an ion channel,
You have to rewrite and recompile the whole thing.
the whole thing, from scratch. In a field like systems biology, where knowledge is constantly changing, that is a crippling bottleneck. Geppetto was designed specifically to allow constant, easy refinement.
And they made some specific software engineering choices to enable that. You mentioned something called OSG.
OSG is key. It enforces what's called low coupling between code modules. Think of it like a smartphone's operating system that lets you install and uninstall apps without having to reboot the whole phone.
Okay, I get that.
In Geppetto, this means a scientist can, say, replace a simple neural model with a much more complex one, or swap out the physics engine for agar with one for water at runtime without breaking the rest of the simulation.
That ability to iterate instantly must speed up the scientific process enormously.
It does. If a model fails to reproduce a behavior, they can quickly swap out the component they think is the problem and test a new hypothesis without waiting days for a recompile.
So Geppetto is the flexible infrastructure. It's the stage, but it's not the actor. It hosts the brain, but it isn't the brain itself.
That's a perfect way to put it. The neurological component, the actual firing model of those 302 neurons, is defined by a different framework called C302. It's a Python-based system that translates the inputs into behavioral outputs.
And I imagine the team realized pretty quickly that modeling all 302 neurons with the absolute maximum level of detail would be computationally insane.
Insanely expensive and maybe not even scientifically necessary. If a simpler model gives you the same behavior, why use the more complex one?
So C302 is built for flexibility.
Flexibility and standardization. It uses standards like NeuroML and LMS. The key takeaway there is that using these standards ensures the models they build are compatible with other simulation tools used in labs all over the world. It maximizes collaboration.
Tell me more about that flexibility. You said C302 allows for, what, 16 configurations of complexity? What does that spectrum look like?
It lets a scientist define a neural network instance anywhere along a complexity axis. At one end, you have the simplest models. Abstract point neuron models like leaky integrated fire. They're computationally cheap and just capture the basics of a neuron firing or not.
And at the other, much more complicated end.
You have these biophysically detailed, highly realistic, Hodgkin-Huxley-based multi-compartmental models. These don't just simulate whether a neuron fires. They simulate the dynamics of ions like calcium flowing through individual channels. They account for the neuron's physical shape.
So they can actually test the hypothesis. Do we need all that expensive, detailed realism to get the worm to crawl, or can we get away with a much simpler, faster model?
Precisely. The answer to that question reveals how much of the worm's behavior is encoded in just the wiring versus how much is hidden in these intrinsic subcellular properties.
Okay, so the neural network fires. That signal has to move the body. That's where the third major component comes in, cybernetic.
Cybernetic is the physics engine. It's built with high-performance languages like C++ and OpenCL. This is where the virtual worm gets its squishiness.
That's a good word for it. It has to model the soft tissues in the environment, right? You can't use the kind of physics engine you'd find in a video game for rigid things like cars or buildings.
Not at all. A worm is fundamentally different. So they use a customized GPU-accelerated algorithm. It's a mouthful. Predictive Corrective Incompressible Smooth Particle Hydrodynamics, PCI-SPH.
Okay, break that down. What's the core idea?
Instead of breaking the worm and its environment into a grid, which is a common approach, SPH treats everything. The worm's body, the water it's in, as millions of tiny, discrete particles that all interact with each other.
And why is the incompressible part of that name important?
Because the worm is mostly water. It holds its volume when it moves. So incompressibility is vital for accurately simulating how forces travel through the worm's body and how the environment, like the thin layer of agar, responds and pushes back.
So let me see if I have this right. Cybernetic gets the motor signals from C302, turns them into muscle contractions, models the bending of the soft body using this SPH algorithm, and then calculates the proprioceptive feedback, the forces and positions of the body.
Which are then sent back through Geppetto to close the loop with the C302 neural circuits for the next time step.
That is the full system, a marvel of multi-scale, multi-algorithm integration. They successfully built the machine.
They did. But having the machine and the blueprint leads us directly into the great, enduring frustration of the project, the functional black box.
This is where the story gets really interesting. We're now confronting the core reason why, after more than a decade of this intense effort, we still don't have a perfect virtual worm.
We have the structural data, the connectome, but the central scientific roadblock is what's called the inverse modeling problem. Exactly. The anatomical map, as complete as it is, only gives you the static wiring. It's a necessary input, but it's completely silent on the dynamic functional parameters. We know who's connected to whom, but we don't know the volume control setting on any of those connections.
I've always liked the analogy of a city map. You know where every road and every bridge is. But the map doesn't tell you if a bridge is closed for repairs, or if a road is one way, or what the speed limits are.
That's a perfect analogy. The function is dynamic, and the simulation requires us to solve this massive inverse modeling problem. We know the desired output, a worm that crawls realistically. We know the components, the 302 neurons. But we have to somehow deduce all the settings that produce that behavior.
And those settings are things like synaptic weights, the exact location and behavior of ion channels.
The background levels of neuromodulators. It's like trying to find a needle in a haystack, but the haystack is 1,000 miles wide.
What's the scale of this search space we're talking about?
It's computationally enormous. I mean, just imagine. You have 302 components, but each one has maybe 20 to 50 unknown parameters, things like the conductance for a potassium channel or the strength of a gap junction. You multiply all those possibilities together, and you have a solution space so vast that you can't possibly brute force it.
You're trying to reverse engineer a complex recipe just by tasting the final cake.
Yes, and the sources are very clear that behavior is driven by these dynamic properties, not just the static connections. One of the key missing properties is how the neurons themselves work.
What do you mean?
Well, unlike vertebrates, which rely heavily on these fast, all-or-nothing action potentials, a neuron is either on or off C. Elegans primarily uses slowly varying graded potentials.
So the signal isn't just on or off, it's a continuous analog voltage level. Why does that make the modeling so much harder?
Because the exact voltage level dictates everything. It determines how much neurotransmitter is released. And that voltage level is exquisitely sensitive to the density and characteristics of all the different ion channels in the neuron's membrane. If your model of one channel is just slightly off, the neuron's entire firing pattern might change dramatically.
And the connectome map doesn't tell you where those channels are or what their settings are?
Not at all. That has to be measured experimentally, which is incredibly difficult, or it has to be estimated.
So the worm's connectome is structurally complete but functionally blind. And there's another major issue you brought up, the same structure, many functions problem.
This is a really deep limitation of connectomics in general. The physical wiring might be fixed, but the functional state of the neural circuit can change on a dime based on the chemical environment.
A neuron can switch roles.
It can, from inhibiting a partner to exciting it, or just change the strength of its output, all based on external chemical signals.
Can you give a concrete example of how that affects the simulation of something basic like crawling?
Sure, let's stick with crawling. The basic circuit for forward motion is fixed. We know the wires. However, the precise rate of locomotion, the speed the worm crawls at, is tightly regulated by neuromodulators, especially dopamine.
Okay.
If the current Geppetto model doesn't include the precise spatial and temporal effects of dopamine, the simulation might produce a crawling motion, sure, but it won't be able to reproduce sophisticated, real-world speed adjustments. The structure is the same, but the function, the speed control, is missing because the chemical context is absent.
And to add to all this frustration, there's even a technical limitation with the original 1986 map itself.
That's right. Because the map was derived purely from electron microscopy, it just shows you where two cells touch. But in C. elegans, unlike in some other systems, those images can't inherently tell you if a synapse is inhibitory, if it shuts down the next neuron, or if it's excitatory, meaning it stimulates it.
So for every single connection, the modelers have to guess or rely on other painstaking experiments to determine if that connection turns a signal up or turns it down.
Absolutely. That lack of polarity information just massively increases the difficulty of tuning the network. It dramatically reduces the power of the wiring diagram to act as a reliable guide.
So let's list some of these crucial missing biological parameters that the teams are working so hard to find.
Well, first and foremost are the specific synaptic strengths, or the connection weights. How strong is this connection? Then there's dynamic plasticity, the idea that those weights can change over time based on the worm's experience.
And this brings us back to that huge unknown you mentioned, long-range neuromodulation.
This is maybe the biggest known unknown. We're talking about neuropeptides and monoamines. These are chemical signals that don't just jump across one tiny synapse. They diffuse widely through the tissue like a mist, affecting multiple neurons at once through volume transmission. They basically set the global behavioral state of the worm.
So if the worm smells a predator, it's not just one synapse firing. It's a global chemical shift that changes the operational settings for dozens of neurons all at once.
Exactly. And modeling that level of slow, pervasive system modulation is extraordinarily difficult. To try and get a handle on the very basics of this, the community launched a sub-project called ChannelWorm 2.
And what's its goal?
It's specifically designed to curate and model quantitative data on the worm's ion channels. They're aiming for biophysical realism at the subcellular level. For example, looking at calcium dynamics in key interneurons. If you get the calcium flow wrong in one central neuron, the error can cascade and break the whole circuit.
So they're working backwards, trying to gather all these crucial missing pieces. But even if they get all the data, there's this unsettling possibility that there isn't one single correct answer.
That's the problem of non-uniqueness of solutions. It's highly probable, given how variable biology is, that there are multiple sets of parameters, multiple different combinations of synaptic weights and channel properties that could all produce functionally realistic worm-like behavior.
Which means if they find one set of parameters that works, they can't be sure it's the actual set the real worm uses.
Right. It just means they found a functional spot in this impossibly vast solution space. You're not searching for a single peak on a mountain. You're searching a vast, bumpy plateau for any spot that works.
And this isn't a new realization, is it? Other projects have hit this wall before.
They have. Earlier modeling efforts, like the Virtual C elegans project at Hiroshima University in the mid-2000s, faced this. They also got simulations of simple behaviors, like the worm pulling back from a poke.
How did they do it without the data?
They used machine learning, specifically evolutionary algorithms, to basically evolve the connection weights until the network performed the right action.
So if machine learning can find functional weights, why is the OpenWorm project so insistent on finding the biologically realistic parameters? Isn't a working virtual worm good enough?
That's the core philosophical difference. If you use machine learning to find any set of parameters that works, the model might work, but it doesn't necessarily explain how the real worm works. The weights you found might be non-physical.
They might not obey known biological rules.
Exactly. OpenWorm is committed to bottom-up systems biology. Their goal isn't just prediction, it's understanding. They want their model to fail in the same ways the real worm fails if you, say, knock out a gene. And you can only get that level of explanatory power if the parameters are biologically realistic.
That struggle for realism is why this has been a decade-plus journey.
And it's what makes the project so scientifically important.
This intense struggle to perfectly mimic reality really forces us to ask, how do you even define success when you're building a virtual organism? Let's move on and look at OpenWorm's validation standards and their pioneering commitment to open science.
Well, the project's guiding philosophy is very pragmatic. They have this saying, all models are wrong, some models are useful. So their definition of a complete model is purely a functional standard.
What does that mean in practice?
It means the model is considered complete when it statistically fits all the available experimental data about the wild-type worm's microscopic behavior.
So they're not aiming for a perfect one-to-one cellular copy. They're aiming for behavioral fidelity. The high-level output of the system has to match reality.
Exactly. The real worm is the ultimate benchmark. They rely on these huge existing data sets that provide quantifiable metrics, things like locomotion speed, how often the worm changes direction, the rate and shape of its turns, like its signature omega turn.
And they have an automated system for this validation, right? The movement validation framework.
The MVF, yeah. It's basically a massive automated testing suite. It runs the simulation, it extracts all the measurable outputs, and it compares them statistically against the data from real worms.
And the beauty of that is when the simulation deviates, say it crawls too fast or turns too rarely, that deviation points directly to a problem.
It does. It immediately tells the scientists that a crucial biophysical parameter is either missing or it's calibrated incorrectly. It turns a scientific failure into a clear engineering directive for the next round of improvements.
It's like turning theoretical biology into hard-nosed quality control. But they also have a more philosophical benchmark, right? Something called the imitation game for biology.
Yeah, proposed by David Harrell. It's a powerful Turing-like test adapted for in silico biology. The setup is simple. A human expert, a top-tier worm biologist, is given two sets of objective, measurable data.
One from the simulation, one from a real worm. And the data is unlabeled.
Right. And the interrogator, the expert, has to try and guess which data set is which.
And the model only passes if the expert can't reliably tell the difference.
Exactly. It's a very rigorous standard because it means the simulated organism behaves so realistically across quantifiable metrics that it actually fools the people who know it best.
And I like that this test acknowledges that success is always moving. It evolves over time.
Precisely. If, in 5 years, we discover some new, subtle biomarker in the worm's behavior, the expert suddenly has a new way to tell the difference, and the model fails the test until that new knowledge is incorporated. The goalpost is always moving, which is as it should be.
Now, beyond just replicating the adult worm, they launched a really innovative sub-project called DevaWorm, which focuses on embryogenesis. Why spend resources on development when the adult simulation is still so hard?
It's a brilliant strategy, really. It's about tackling that inverse modeling problem from a completely different angle. The rationale is that understanding the process of how the adult nervous system was built can give you crucial constraints on those unknown parameters.
So instead of just guessing the density of an ion channel in an adult neuron, you try to model how that channel profile emerged during development.
Exactly. Embryogenesis is a systematic, highly constrained process. The DevaWorm team models how the nervous system develops, and from that, they can infer crucial constraints on the adult properties.
And they're using some really advanced techniques for this, right? Things like developmental graph neural networks.
DGNNs, yeah. And also something called hypergraph analysis. In a normal graph, a connection just links two things. But in biology, processes are more complex. A cell's fate might depend on three of its neighbors and a chemical signal all at the same time.
So a hypergraph can model those multi-way relationships.
Yes, it allows them to model these complex developmental processes and look for the global top-down rules that influence the final adult structure. It provides constraints that you just can't get from studying the adult alone.
It sounds like DevaWorm is suggesting that maybe you can't fully model the adult unless you first model how it was built.
That's the essence of the hypothesis. And this whole massive operation, Geppetto, C302, DevaWorm, all of it, is held together by one key operational mandate: open science.
Right. The OpenWorm model is almost a direct response to the problems you see in traditional siloed academic science.
It is. Integrative biology often suffers from data fragmentation, where one lab's data is incompatible with another lab's tools, and from a lack of reproducibility. It creates a lot of waste.
And OpenWorm mitigates this by enforcing transparency. Everything is on public GitHub repositories with open licenses.
This does a few things. First, it democratizes access. Students, engineers, scientists from anywhere in the world can contribute. Second, it enforces standardization and continuous peer review through the open development process. The whole project is far more resilient than one relying on a single, massive grant.
It's really proof that a big science project doesn't always need a multi-billion-dollar centralized budget if it can leverage the power of open source collaboration. And that brings us to the conclusion of our deep dive into the worm that no computer scientist can crack. We've seen this phenomenal engineering success paired with a relentless scientific frustration.
They certainly built the platform they needed. The architecture is robust and modular. Geppetto integrates everything. C302 handles the neural modeling. Cybernetic provides the soft body physics. They've successfully closed that critical neuromechanical loop.
They've achieved functional simulations of the basic components of movement.
They have. They can generate the traveling waves of muscle activation needed for forward crawling. But the persistent roadblock, that black box, remains those dynamic functional parameters. The project is still stuck on the transition from simple movement to complex, context-dependent behaviors.
Like the tap withdrawal response.
Exactly, a classic worm behavior. A crawling worm senses a mechanical shock and instantly starts moving backward. That requires accurate sensory input, intermediate neurons firing correctly, and the precise, instantaneous reversal of the entire motor pattern.
And that reversal depends entirely on those functional parameters, the correct synaptic strengths, the balance of excitation and inhibition.
All things that are largely missing from the anatomical connectome. If the model can't reproduce something as fundamental as the tap withdrawal, it confirms they're missing something crucial. Maybe it's plasticity, maybe it's the global effect of neuromodulators like serotonin or dopamine.
So the ongoing pursuit is really an intense tuning effort.
It is. They're using parameter optimization techniques, machine learning, not just to find any solution, but to search for functional synaptic strengths that fit known biological constraints and produce realistic activity plots in the neurons. The belief is still strong. The physics and the biology we know are sufficient if only they can find the correct, biologically realistic settings.
The stakes are incredibly high. If they eventually succeed, OpenWorm will be an unparalleled tool for scientists. A complete organism model to perform experiments in silico, testing hypotheses about mutations or drugs before doing expensive lab work.
But here is the final provocative thought for you to chew on. If the simplest nervous system in nature with a fully known, complete anatomical map can resist digital emulation for more than a decade, what does this tell us about the limitations of pure structural data?
It suggests that the key to understanding a brain isn't just the component list, it's the secret recipe hidden in the parameters that govern how those components interact.
And it raises this profound question. What essential dynamic principles of life principles related to emergence, to chemical state, to context-dependent function remain uncodified in our understanding of biology? The struggle to crack this tiny 1 millimeter worm might just be revealing the sheer scale of the knowledge we're still missing about every single brain, including our own.